happy to welcome everybody to this evening's ELSC seminar. This is the last seminar of this year, and today we have a very special guest. Our guest today is Nachum Ulanovsky, who graduated in physics in Tel Aviv University and then pursued his PhD here with our very own Eddie Nelke, working on some surprising features of the auditory cortex. And he was looking into processing of low-frequency events by the auditory cortex as part of its elementary function. This was still in the time when Eddie was in the remote in Kerem campus. And in 2004, he went to the University of Maryland for a postdoc in the lab of Cynthia Moss, where he first started working on bats and their truly unique navigation and localization systems. This sounds already a little bit complicated enough for the ordinary person. Nachum decided maybe it might be more interesting to make it a little bit more difficult to do it in behaving, echolocating, and navigating bats. And this resulted in a number of interesting and intriguing insights on the network activity of the bat hippocampus during exploration and navigation of its environment. In 2007, he started his lab at the Weizmann Institute, continuing to work on the navigation and echolocation and freely moving bats, where his lab members discovered some of the most amazing findings about the representation of the bat's three-dimensional space, which I hope we will hear more today. Thank you very much for accepting the invitation.
I want to say, well, I'm not Nachum. No, I'm not Nachum, but I will say one word. First, I want to say the word that every time that the graduate of the ICLC PhD program at the time, which is now ELSC program, comes and gives a lecture. It's a special honor and pleasure to have Nachum, because I was sitting very close to his lab, seeing how things progress, and I knew that one day he will be a member of ELSC. And you should have said, unfortunately, he's a professor at Weizmann, but it's okay, we accept you as well. And then the second part is that I have to apologize to Nachum and to many of you that as the director of ELSC who knew the program of the seminar, we didn't notice that we invited him for the last seminar, which is just 2 days before next week. And you know that next week is the big week. You all know it, yeah? Right? Everyone is registered. Everyone will be there. See you next week, and now.
So, hello everyone. Do you hear me at the back? So it's great to be here, and it has been I think 7 or 8 years since I last gave a talk at the seminar. Here. So on one hand, I had to think hard what will I skip because it's impossible to cover in 1 hour everything that we did since I arrived to Weizmann Institute, which was 8 years ago. On the other hand, because of this unfortunate mistake that next week I'm giving a talk in the conference, I just decided to take advantage of it and decorrelate at least partially the talk. So today and next week and next Thursday I'll talk about partially different things. There'll be some overlap. I want to say 40-50%, Ellen said say 35%, but certainly there'll be a major story I'll tell about today that I'm not going to talk about at all next week and vice versa. So 40% decorrelation. So the title of my talk is Neural Code for 3D Goal-Directed Navigation and the Hippocampal Formation of Bats. Now the first part of this sentence is a bit more vague. Chaim just told me I came to hear what's natural neuroscience. I know what is unnatural neuroscience, and I answered I know too what's unnatural neuroscience, most of it, but I'm still looking what this thing is, and what I'll try to convey today is my belief, and at this point it's mostly belief, but there is some evidence for it, that the more you go to the natural behaviors of the animal to which the brain evolved to function, you'll find surprising things about the brain. And primarily both for conceptual reasons, but primarily for technological reasons, neuroscience for many decades was away from that, doing either very simplified scenarios, very simplified behaviors that are far remote from natural behavior, and I'm not there yet. But we're moving slowly in that direction, and I think today's talk will be a little bit the story of our journey towards that goal. So no answer, it's mostly a question. So we're not working understand the entire neural basis of behavior, which is eventually my interest, but neural basis of spatial behaviors, spatial cognition, spatial memory, and navigation. And we work on the hippocampus and frontal cortex. So the hippocampus, this is in a rat brain. The hippocampus is this sausage, and the frontal cortex is this strip at the back of the brain. And they're known for many years to be important for memory in general, but also in particular for spatial memory, spatial cognition, and navigation. And in those brain areas, I'm sure you all know this, people have found in the past very interesting cells. So John O'Keefe in the early 70s discovered place cells. So if you let a rat run in a square box, let's say 1 by 1 meter in size, so the rat is running around, the black line is the path of the animal, the red dots are spikes, and you can turn it into a heat map, so you see the neuron was active about here. So this is what we call the play field of the neuron, and this is the play cell in the hippocampus. In the frontal cortex, one synapse before or one synapse after the hippocampus, depends how we look at it, it's a loop. There are grid cells, so these are neurons that are active in a set of positions in the environment, forming a hexagonal lattice. And there are also head-direction cells, neurons that don't care so much about where the animal is, but more about its direction in space. So you can think of it as possibly a neural map, perhaps a way to measure distances, and a compass. So all the elements you want in order to build a navigation system in the brain. We studied these types of cells in bats. In fact, I'll defer to the end to explain why bats. I think it's easier first to go through what we're doing, and then I think it'll become clear what are the advantages of bats. But I just want to say, we're not in the business of understanding bats, although they're cool animals. We're in the business of understanding the brain. We are trying to use bats as an animal model to understand the brain. So this is kind of the question which relates to natural neuroscience in the context of the navigation system. So how does real app navigation differ from navigating in a 1 by 1 meter empty box? Most of what we know about these brain areas come from these kinds of experiments. And you can think of many ways. I can add at least a couple of major issues here, but I'll put just 3 here. First of all, the world is not flat and flat boxes. It is complex, it's multi-compartments, and it's three-dimensional. So we've done a lot of work about three-dimensional representation in the brain. I'll tell you about this today. Also, the world is not empty. It contains gold. I just told you that place cells and head-direction cells, etc. What do they tell me? They tell me I am here, and this is north. It doesn't tell me at all. Let's say I want to go back after this lecture to the Weizmann Institute. Where is the Weizmann Institute on the map? We'll say it's not on the map at all, but it's not in this conceptual framework that I just told you about. There's nothing about goals. And to be provocative, you don't even need to know your place, where you are and where is north. All you need to know is where is the Weizmann Institute, where to get there. So how are goals represented in the brain, I think, is a major issue, and I'll show you some data about that. And also, of course, the obvious thing, the world is not 1 by 1 meter, and navigation does not occur on these distances, but in kilometer scales, and we're starting to address this as well. Yes?
My best of my mind is basically shows that they are goal related. So
you mean the
later. So if you switch the goal to nothing else, the places also.
I'll get to that. The answer is no, but I'll make more specific what I mean here. Okay? Just to put the framework of kind of what I mean by going to more natural behavior to understand how this works out in the brain. So this animal we worked on, the Egyptian fruit bat, it's, well, first of all, just by looking at it, you'll immediately see it has big eyes, so it's not blind, unlike the same blind as the bat. Not true. There are 1,300 species of bats in the world, 25% almost of mammalian species. None of them is blind. These guys see really well, much better than a rat. Their acuity is almost about half of a cat, so they really see well. They also have a sense of sonar. I'll say a word about this in a second. They're also very big, at least by bat standards. 150 to 180 grams means practically they can put on them a lot of equipment. You can put neural telemetry systems. We developed GPS data loggers, a lot of weight that bats that live in North America and Europe that are 3, 4, 20 times smaller cannot carry. So we have kind of a size advantage, and when I came back to Israel, I switched from a 15-gram bat to 150-gram or 180-gram bat, kind of 10 times larger. It's a low-tech solution to the miniaturization problems. We can do these bat experiments that are impossible in other bats. And what do we typically measure in the lab? We have a big flight room. Actually, we have 3 flight rooms, 2 small ones and 1 big one, 5 or 6 meters, and we have multiple cameras, and we can reconstruct a three-dimensional position of the animal in space. We can also record its echolocation missions. The bat emits sounds ultrasonic sounds that travel in air and return to the animal, and by the time difference of arrival, it can compute distances and can compute directions and object shapes and a lot of other interesting things. We can also, and we don't do it routinely, we've done a study in the past where we've looked at how is the animal steering its sonar beam in space. And so, well, this is the cartoon bat, but the beam is real. This is how emission looks. It's directional. It goes mostly in this direction, tapers off, and we're using a microphone arrays and two cameras. You can ask how is the bat directing its beam in space, and you'll see a bat, just an example, a bat flying from here, landing on this ball, and these are the top view. These are the microphone array, bat flying, landing here, and the magenta curves are the emissions. So you see it's directing its beam very repetitively, left, right, right, left, left, right, right, left. And as it approaches the target, you'll notice it seems to try not to direct its beam to the target. So very carefully look to the right, to the left of the target. And this was the first study that we published from the lab 5 years ago. And the rationale that we came up with explaining this behavior is the following. So if this is the emission power versus azimuth, and this is the direction to the target, so if you put, but most people would think you put the maximum energy on your target, so you get maximal echoes. This is good for detection. You maximize signal-to-noise ratio. On the other hand, this is the worst place for localization, because if the target, the relative angle between you and the target now moves slightly, this is zero derivative here, there'll be no change in intensity. If you want to use that cue for localization, actually you want to look to the left and to the right of the target and put the maximal slope of your emission on the target. And we showed that exactly that they're not just randomly looking left and right, but exactly putting the maximal slope on the target, maximizing spatial information. So it's a mathematically optimal behavior for localizing direction of target. As another way to put you into kind of the world of bats, I want to tell you another study that's already a bit old, 4 years ago, where we asked how do bats navigate in the wild. In fact, it was at that time a question about how mammals navigate, because this kind of thing was not done in any mammals before, meaning tracking with the GPS, so having high resolution information about the way they navigate. So with colleagues in Italy and Switzerland, we developed this GPS data logger. So it just stores the data on board a flash chip there. Now we have a smaller device made by an Israeli company. And we just, this was done in collaboration with Weizmann here at the Hebrew University. And we put the glue, with medical skin glue, the GPS data logger on the back of the bat and release it, and And you'll see that the bat here went from its cave. This is not a wild type bat, this is a wild type, a wild bat, literally. In a cave, kind of halfway between here and Rehovot. So the bat leaves its cave, goes to a local tree for a few minutes, it's a fruit bat, and then goes to these two trees. You see the scale here, it's 5 kilometers. It's about almost 20 kilometers. He feeds there for the entire night, and then slides extremely straight back to the cave. So these guys can really navigate long distances, and it's not just this bat, but now with smaller Israeli GPS, we can now record multiple nights, actually up to 75 nights now. And this is an example of all this data. Is one animal, each line is a different night, and you see that this individual bat flies from the cave to the tree and back and forth to the same tree, you know, by a flyway. It's not exactly reproducible flies. You know, it can fly a half a kilometer to the right, half a kilometer to the left, but reasonably reproducible, and going to the same individual tree night after night.
Is there just one tree?
No, there are multiple trees, and I'll get to that later on. We'll talk about goals, because actually we have some answers to that. Of course, eventually fruits get depleted, so they have to switch to something else. I'll show you how that happens. And again, I'm skipping a lot of details that were in this paper, but we did also a lot of multiple manipulations. We released them in distal locations and show that the vision is important for this kind of long-range navigation. But echolocation is useless because it's a short-range system. It cannot work over many kilometers in physics. So with vision, they have very good vision, and by releasing them in a big crater in the Machtesh Gadol, they basically got lost, and we released them on a high mountain near the crater from which they can see the center of Israel. They flew straight. So by this manipulation, which mainly affects the visual input, I think we provide at least some evidence that vision is important for this kind of navigation. Okay, so this was kind of an introduction to the world of bats, and this is what I'm actually going to tell you about today. I'll tell you about spatial cells, about theta oscillations in bats. Then we'll go to 3D, kind of more natural 3D. This will be mostly in kind of two-dimensional conditions. This is a three-dimensional, I'll tell you about three-dimensional place cells, 3D grid cells, 3D border cells, and 3D direction cells. I'll tell you about goal-directed navigation using stored representation. This is the very new data, not unpublished, it's not close to being published. And I'll tell you about some future plans, not exactly future already, closer to present, but anyway, about large-scale kind of 1 kilometer representation. Okay. So the first story is a study that was started by Michael Yartsev and was continued by Tamir Eliyahu, 2 graduate students in the lab. Michael Yartsev is actually now visiting the country in a few weeks, starting his own lab, his bat lab in Berkeley. So basically, just to start off, I'm kind of summarizing here several years of work to tell you that in 2 dimensions, the story in bats is most of it is similar to rats. That is, that's like I showed you, there are place cells. There are grid cells, there are head direction cells in rats. There are also border cells that I didn't mention, but these are neurons that are active along the borders of the environment, sort of encoding the geometry. We find the same kinds of cells in bats, place cells, grid cells, border cells, and head direction cells. And most of their properties were similar. There was one property that right from the start, even when during my postdoc, was obviously very different, and that is what's called the theta oscillation. So theta oscillation is the biggest in terms of just voltage. If you stick an electrode in the brain, it's the biggest electrode anywhere in the brain of a rodent. And this is how it looks when you record in a rat. And this is from the Zaki lab. You see a very periodic, about 8 Hertz oscillation in the rodent brain. In bats, and by the way, also in humans and monkeys, it doesn't look like that. It appears, theta oscillations appear very intermittently. So you'll have maybe 1 or 2 seconds of an oscillation, and then it will go away. Very aperiodic. I'll show you later some more LFP traces. It seems very aperiodic and nothing close to this periodicity. Now, why is that important? I mean, it's important for a number of reasons about which I'll talk later, but to this study it was important because of models of grid cells. So there are 2 major classes of models of grid cells. And I won't talk about them in detail, but one class, there are dozens of studies that can classify most of them into these kinds of models. One kind of model is a network model. Basically, you assume a Mexican has connectivity, and then you have on the sheet of the neurons, you have the hexagonal structure forming, because that's the way the shortest distances between the blobs are fixed on the network, and there are various associations of that. Now you need to move this activity bump with the movement of the animal, which is non-trivial, so you need to introduce some symmetry in the synaptic connections, and the model took care of that, and this gives you the grid structure. So essentially, the grid structure on the sheet of neurons kind of falls out, I wouldn't say automatically, but almost by itself from the circular symmetry of the connectivity. Okay, so that's one kind of model. Then there's a completely different kind of model, which relies on oscillations. So basically the idea is that you have these theta oscillations that I just told you about in the soma, and there's some of these models that you have in the dendrite a different frequency of an oscillation, that the frequency difference is proportional to the running velocity of the animal. So then the model say you get a beat between these 2 oscillations. Now if the animal runs twice as fast, then the frequency of the beat will be twice as high, and then the period will be half, but because it runs twice as fast, the wavelength in space will be fixed. Okay, so by this assumption, the frequency difference is proportional to the running velocity, you created a fixed wavelength in space irrespective of running distance. So you kind of integrated the movement essentially. The problem is, or was, that in rodents it's very difficult to dissociate these 2 models, because at least under normal conditions you always see the grid cells and those theta oscillations. So, and if you do manipulations, then you always affect other things. So there were major debates, but it's really difficult to dissociate these things. But what I just told you about that is that at least in the hippocampus, which is one side of the stack, we see place cells without theta oscillations. So then we reasoned with Michael that if we go to the entorhinal cortex and find their grid cells without theta oscillations, we basically rule out these kinds of models. This is what we've done, and after hard work, Michael was able to record grid cells in the entorhinal cortex. And so this is an example of grid cells from a bat, a crawling bat. And what about the oscillations? These are kind of the key question here. So if you look at a mouse brain, you stick a nose with into the entorhinal cortex, look at the power spectrum for local field potential, you get this huge peak at around 8 Hertz. This is the theta oscillation. If you do the same in the bat, you get nothing of that kind. Something like 1 over F dependence, but no discernible peak at any frequency. If you look at the spike rate autocorrelations, they're very periodic, very smoothly periodic in the mouse. If you look at the bat, it looks much less periodic. Nothing pops out like this. How do you quantify the significance of that? So basically, we did what people have done in rats, which is to compute the power spectrum of the autocorrelation and normalize it in some way. This gives you a theta index, and then you shuffle 1,000 times the spike train, and this gives you the confidence interval, and you see that none of the real data in blue fell outside the confidence interval in gray, which means there are no significant oscillations at theta frequency in the bat. And we concluded from that theta oscillations are not necessary. So this model basically is not true, right? Because this model says you need A to get B, you need an oscillation in time to get an oscillation in space. If you have an oscillation in space without an oscillation in time, B without A, so you'll logically rule this out. Okay, so this was published already. Now, there were several critiques raised against our findings. One is that the firing rates of grid cells in crawling, and actually play cells as well, in crawling bats tend to be relatively low. So maybe there's something about the statistical power, and it's not true. But anyway, this was one issue, and I can already tell you that in flying animals, flying bats, firing rates go high dramatically. But for crawling data, this was one argument. And the other argument was maybe it works at different frequencies. There's nothing magical about 8 hertz. Maybe the principle is true, but for whatever reason in that it's 3 hertz. Okay, so we should look at other frequencies. So this is what we've done. We now look at all possible frequencies on unbiased manner, and in particular we focus on interneurons. Why interneurons? Interneurons are in the hippocampal formation. First of all, they're called theta cells to this day because they're extremely oscillatory. They're even more theta modulated than the pyramidal cells. So it's kind of the ultimate test bed. And secondly, they're extremely high firing rate. So if you don't see oscillation in interneuron, it's probably not there permanent, for real. So this is how autocorrelation looks in rat neurons, for the motors in this case. And this is how a couple of cells look in the bat. You see that the autocorrelation is flat. There's really nothing there. These are 5 more examples. Really flat. I mean, this looks like it's done with a ruler. No, it's real data. So it's really, really, really flat. There's no oscillation just by looking at it. Okay, so now we can take each of these autocorrelations and stack them above each other. So now each line is an autocorrelation color-coded. So we have 409 cells from the motors, and we have our cells in the bat. Now from all data sets, we have interneurons, in flight, in crawling, plate cells, and grid cells. And basically, if you look at the rat, you see this vertical strike at about 125 interval. This is the 8 hertz theta oscillation. In the bat, there is no vertical band that you can see. If you had something, let's say at 2 hertz, you'd see a band here at 500 milliseconds, but there's nothing that at least pops at you by looking at it. But it's just looking, maybe there's still something there somehow. So you need to do some testing of that. So we've done it 2 ways. One is we've fitted a decaying sine wave. This is essentially the red line. And so for the rat data, we'll use that to compute an oscillation index. The oscillation indexes for the rat data are very high, and all the frequencies that we extract empirically. What is the best fitted frequency? And in the rat, as you might expect, that has been seen for since 1935, so 80 years. You see this 8 hertz oscillation. Now, if you do the same for the bat, the oscillation indexes are much lower, and the frequencies, some neurons, which are the dots here, are significantly oscillatory, but the frequencies are all over the place. Maybe it's noise. Okay, and then we did another kind of analysis, which is spectral analysis, the way it's typically done, which is to look at the power spectrum of autocorrelation. This is how it looks in the rat. This is the peak at 8 hertz, and by the way, these are small, just a few hundred spikes, similar numbers of spikes as in the rat. So I don't agree to the statistical argument. We have similar numbers of spikes for the rat, but still, it doesn't look at all as here. No peak at 8 hertz or anything clear at other frequencies. It looks very messy. And again, a few cells pass the significance criterion, but they're all over the place in terms of frequency. And now we do the critical comparison, which is to ask which neurons are significant using both the spectral and the temporal analysis. So in the rat, both analyses capture about 70% of the cells. Most cells get captured by both analyses, and the frequencies are the same. As you follow the diagram, the frequency is one method frequency using another method. In the bat, literally 0, 0, 0, and 0 neurons are significant by both methods. So I think this is kind of a definitive demonstration that there are no oscillations at any frequency in the bat. So our conclusion is that the analysis of in vivo data from bats does not reveal movement-related oscillations at any frequency, neither at grid cells, nor plate cells, nor interneurons, not in 2D nor 3D flight, and that this argues against the notion that a filter interference plays a role at the system for grid cell generation. Yeah, this was the story until a few weeks ago. Now there's a twist. And this is the first time I'm talking about it, so I'll be happy to answer. So, okay, the title says it all. So, so this is how, let me just show you this trait. This is from a rat. Okay, I have this very periodic oscillation in the rat. Now, this is how it looks in the bat, okay? This is a crawling bat in this case, yeah. Now we're starting to collect LSP data in flight. It's tricky, and we didn't even have a technical ability to go to that low frequency until recently, but now we're able to do it. But most of the LSP data is in flight. We have some in flight. So we see sometimes its period is short, sometimes it's long. It's kind of much more messy, nothing similar to that. But now the twist that happened is that we asked ourselves, okay, for the for the filter interference model of grid cells, it's critical to have a perfect sinusoid because it really relies on a sine wave to get perfect sinusoid in space, which is the grid. But for a lot of the other functions of the hippocampal formation that use the model that's used as oscillations, if you look at the model, the oscillation is not important. What's important is co-modulation, in most cases, co-modulation of neurons. You want neurons to fire together, to be modulated together. So then we ask ourselves, what if, let's say, this is a cartoon of what happens in the bat. The frequency just sometimes high, sometimes low, it's kind of all over the place. But what now in the rat? I forgot to say, in the rat, neurons, pyramidal neurons in the hippocampus are phase locked to the trough of the theta oscillation. So a little bit after the trough, about 60 degrees after the trough, 60 degrees in space. What if the same thing happens here? Okay, let's assume that this aperiodic thing still synchronizes the neuron, and they still have phase locking, which is the red, the red spikes, all the cartoon, of course, that phase lock around the trough, or maybe a little bit later. So then synchronizing populations together will still work just with using an aperiodic synchronizer. So we looked at that starting a few weeks ago, and quite amazingly, we see that now if you compute the phase, you can still compute a phase out of this thing, right? You need to do some filtering, but essentially you can say this is one period and this is not this, 360 degrees and this is also 360 degrees, etc., right? It just varies, but you normalize everything to phase, okay? So now this is this very aperiodic phase. And this is examples of two neurons, one from C1 and one from MEC. And what you see in these examples is that these neurons are quite periodic. They would not come out periodic in the regular autocorrelation because the regular autocorrelation, the period just goes all over the place. But if you look normalized to phase, it turns out they're phase locked. These are two examples, and I can tell you already just by looking at examples, we are still working on the population analysis. There are a lot of them. It's not a rare example. So this is basically kind of where we stand with this little twist. And so I would still say that what I just told you is true, that for the grid generation, I still think we killed that model. But another implication is that for most other models of why you need a theta oscillation in the hippocampus, my argument would be that what you really care about is not oscillations, but synchronization. And this is exemplified by these data. And there's some indications that the same happens in humans. That you don't have oscillations, we know that, but there is phase locking there, etc. Yes.
Isn't it obvious from the fact that it has LSP oscillations, modulation?
Yeah.
It doesn't come from...
Hi, you're smarter than I. Yeah, I could have thought about this earlier, but the fact that LSP... You're right, the fact that LSP is not flat... Right, you're right. The fact that the LSP is not flat means that there is something there, so yeah. It took us a number of years to get there.
You also showed us an example in the beginning where there are theta oscillations in the bed.
Right, so it's very... So this is what we're trying to understand, the spectral-temporal structure of that. Is it a narrow frequency signal that, in the rat, it's just, the spectrum will be very constant, in the bat, it's just all, it wanders, it's a narrow frequency, momentarily narrow band, but it wanders around, or maybe it's something more complex. And I don't have an answer for that yet. So you see examples of that, you see examples of this. So, but you see, that analysis was fixated on theta. Here we are now, don't care about the frequency, we just focus on theta. Yeah.
The first question is regarding how you lock the neuron for the LSP. So if you look at correlation between the LSP and spike.
This is called LSP to spike. So this is, sorry, maybe I wasn't clear enough. This axis is two repetitions of the phase of the LSP.
Okay. So the logic. And second question, do you have any idea about the distribution of the preferred phase?
This is what we're looking at. It looks like there is some peak after 0, which actually numerically is the same average phase as in rats, but we're still looking at that. Because there are issues of how you filter. It's more difficult, I can tell you, to compute the phase here with these noisy data, so it's more tricky, so we're looking at that. So I don't want to say, yes, it's the same kind of average phase as in rats, but certainly I can tell you that whatever way you filter it, we see a lot of neurons. I'm talking about dozens of percent that look like that, whichever way you do it. So, yeah, this is what I know so far. No, it's way too early. You can't do much into it. Yeah.
So what about phase precession?
Okay, this is for the aficionado. I can tell you that we do see phase coding without oscillations, at least in some neurons. So we do see this nice grid cell that I showed you, this neuron and a few others that we already looked at, do show phase precession in the sense that when you, as you move through, so phase precession, for those of you who don't know, is that not just that the spikes occur at a fixed phase, but they go at an earlier, progress an earlier and earlier phase. And we see that now with periodic phase, at least in some neurons, including this beautiful grid cell. So, but as I said, it's very, very, very early. Yes?
Usually don't have the back, so I don't answer. But I mean, has to do something with the fact that they move in a constant, very gradient rate or something, but they're crawling, not their natural.
Yeah, so I thought that it's due to the crawling for a number of years because of the way they move, and maybe because also the sensory systems, because rats are I always thought it's because of the behavior. Rats are extremely periodic animals, right? They have the whisking and the sniffing, which we now know from David Weizmann controls the same oscillator in the brainstem, and everything is very periodic, and that's actually phase lock to the phase oscillation in hippocampus in many cases. And in the bat, when it's crawling, there's nothing like it. But then when they start to fly, and most of the data I showed you are not from flight, the bat becomes as periodic as the rat. The wing beats, the echolocation, the respiration, and in our bats, even the ear movement, are all phase locked and all at 8 Hz. Not just a filter, it's phase locked to each other. So, and still no phase. So, I think it's not that. And I can speculate, maybe I'll leave to the end some speculations. What are the differences? I can tell you in humans it's the same thing. It's very aperiodic, or in monkeys. For 40 years people tried to find nice data in monkeys and failed. And my argument would be, not you personally, but people should stop being fixated on this oscillation. For whatever reason, it's very oscillatory in the rodent. It's aperiodic like this in humans, monkeys, and bats. But what's important is synchronization. So maybe after all, the similarity between the systems is bigger than we thought.
Or maybe equally unimportant.
Or maybe equally unimportant, yes.
It's possible that maybe we have many frequencies of oscillation and no one only predominant, but
Yeah, so we're looking at that now as well. Yeah, it's all, I don't have a lot of answers to these good questions. I'm just showing you ultra preliminary data literally from a few weeks ago. Okay, let me move on. Okay, another story I wanted to tell you about is about orthogonal hippocampal maps for different sensory modalities in flying bats. This is the work done by Maya Geber-Sagiv, who many of you know because she's a student at ICMC. And this is the work that actually hopefully will be accepted for publication soon. But I will not talk about this today, as I promised. I have to skip something, so I'll keep you on your toes. And you'll have to come next week to my talk at the Brady Days conference on Thursday morning. I'll talk about Maya's results, which are very exciting. Okay, now and next week I'll not talk about oscillations at all. Okay, zero oscillations. Okay. 3D spatial codes in the bat brain. This is the next topic I want to tell you about, and this is work also started by Michael Yartsev, and I've been looking at Gilligan and Stalin and Steady Finkelstein on different kinds of cells, 3D grid cells and 3D direction cells. So the basic question is, how will 3D volumetric space be represented by place cells? So if you see a place cell that fires like this in a certain position in a two-dimensional box, you could imagine that in three-dimensional space it could be a sphere, or it could be a cylinder, meaning there's no sensitivity to the height of the animal above ground. I mean, you really don't know until you do the experiment. So we spent a number of years to develop a telemetry system. Actually, it was developed already when I was in my postdoc. A system that allows to record wirelessly spikes from single neurons in flight. So here you see the 4 channels of one tetrode, and here you see spikes recorded in flying back. And so this is took us several years to do that. And the other problem we had to solve is how to train back to cover two-dimensional space nicely and evenly. And I won't tell you about all the details, but we have now good methods to do that because animals tend to have repetitive routes in space. So we basically have to have training tricks to prevent that, and we have those now. And so we put this artificial tree, if you will. For the ethologist, this will be an artificial tree. For psychologists, it will be a three-dimensional 8-arm maze, but it's physically the same thing. And then the animal basically looks for food that's randomly placed on these arms and flies around and around in search of this randomly placed food. And we combine the two, the path of the animal and the spikes. You see that this neuron is active in a delimited region of space, of x and y, but also of z. Okay, and it's not columnar, and in fact 91% of the cells were not significantly different from a sphere. The places were not significantly different from a sphere, meaning that the resolution in the x and the y and the z dimension were the same. It's in the hippocampus. This is the sincere area of the hippocampus of flying back. Okay? So we call it isotropic. It's an isotropic kind of representation, the same resolution in all directions. Again, not all cells, but 91% of the cells. And at least under these conditions, 3D plate fields tile space uniformly with this tree, and we moved every day. We moved the arms of the tree kind of randomly, basically to prevent the bat from developing habits of approaching, certain arms in certain directions. I can already tell you that this is not a general rule. If we now put goals or targets that are important in the room certain positions, then there's overrepresentation developed around those areas. So this uniformity really has to do with a particular behavior, but the isotropy we still see it.
Specificity, how fast is it?
We don't know. We are looking at it now. Yeah. Actually, we're doing surgery on Monday with another project which will partially answer this question as well, but I don't know that. What about grid cells? So this is an ongoing project. We still don't have full answers, but okay, you can say this is the 2 dimensions, optimal packing of spheres. Then in 3 dimensions, will you get something like that, or maybe you'll get a vertical column? Okay, open question. And I don't have full answers yet. This is a project by Gillig Nosal, a graduate student in my lab, and this is an example of a cell recorded by Gillig, and what you see here is that first of all, it looks very different than a place cell I just showed you. Okay, it doesn't have this.
Now in this time.
This in Toronto, we have in Toronto and Pacific. Most of the data is from Toronto. And actually recordings are going on right now on another Toronto animal. And so what you see here is that you don't have this one nice blob of activity like in the place cell, which is very easy. You just look at it and immediately you see it's a place cell. Here, it's very different. You have these multiple blobs. And the other thing is very clear from here is that it's difficult to figure out what's going on just by looking at it. I mean, it looks kind of periodic, but, looking doesn't help much. So, it helps partially, but it's confusing. So what we have to develop are ways to analyze. Do you have a lattice-like structure here? And we don't have full answers yet, but I'll just tell you where we stand now. And so one way that we approach this problem is to say, let's assume it's a certain kind of lattice, and we can now go through all possible, various possible lattices. Let's assume that one of these perfect sphere packing lattices, the FCC, face-centered cubic, which has this tetrahedral-based structure. So it has 4 symmetry planes. Okay, you cut through it in one plane, you get this 2-dimensional hexagonality. You cut another plane, you have a 2-dimensional hexagonality. There's a 3rd plane and 4th plane. So 4 symmetry planes. So now, what you could do is take this, this is a simulation just to illustrate the point, but you take your data and you cut a plane, you do the autocorrelation, you cut a plane through it, and now it varies angles, varies azimuth and varies elevation. and then you plot the two-dimensional gridness, some index of hexagonality versus azimuth and elevation of the plane that cuts through the data. Okay? Now, for this FCC, what you see are 4 hotspots. The color is the how much hexagonal it is. You see 4 hotspots, which exactly correspond to these 4 symmetry planes. Okay? There are 4 combinations of azimuth and elevation where you get high hexagonality. If you look at columns, perfect columns, you get just 1 symmetry plane. Okay? Think of this as Greenland and Antarctic. This is a sphere, so really it's only 1 block. This and this is the same thing, right? So on a sphere, it's really 1 block. You have 1 symmetry plane. As you'd expect for a cube, which I'm not showing you, we have 3. So it's all pretty predictable what you get for perfect lattices. This is for this neuron I just showed you. And you see, it's not exactly like this. But you have, but these 3 seem pretty similar to that. This 4, went somewhere. You have this blue, the low values here and here. So it's not exactly like this, but it's a bit like this. It's only very different than the columns, very different than the cubic, and very different than random blocks, which look basically like random nodes like here. So we see some cells that look like that have a hint that they have some periodic lattice. However, we do see also other cells that have elongated structure. But if you look at the autocorrelation, like this one, this is now a bit of a zoo, what I'm showing you. We don't have answers. I'm just showing you a few examples of different kinds. This is another animal in the zoo, these stripy zebras, okay, the animals that have these elongated structures that certainly don't look like the previous FCC. We also have neurons that look like three-dimensional border cells that are active along, in this case, the floor of the room. We had some on the walls. We had even some crazy cells that look like border cells, like on 3 walls of the room, but they don't cover it evenly, but cover it with something that looks like a 2D grid, so like it's bordered by grid, like a tapete, whatever. So it's a lot of animals in the zoo. Don't have clear answers about this yet, but just showing you some of the things that we see so far. And the, yeah.
But how do you know that there's no, you just use the previous page, but there's no distortion? That's what
You're very right. So now the issue of distortion. If you have distortion, then this large scale measure of that I just showed you will not work. So that's why we also look at local distances. What is the distribution of distances to the nearest neighbor blobs? And I can already tell you it's much more, so this coefficient of variation of the distribution is much smaller than for distribution of random blobs. We also look now at spectral methods, so basically doing the Fourier, look at the Fourier space like in crystallography. We're shooting a lot of guns at this, and I don't have clear answers, but you're right that conceptually it could be that there's a local scale, but not global, and we're looking at that now, and I think there is, based on this distribution of local distances. Okay, the next part of the 3D chapter, if you will, is about 3D head direction cells. So this is work done by Fanny Finkelstein, a graduate student in the lab, and Dori Devdekman when he was a postdoc in the lab at the time, now he's in the Technion. So the basic question that we asked is, are there two-dimensional head direction cells in the bat, right? So I already showed you that you have two-dimensional azimuthal head direction cells in rats and also in bats, but what about 3D, right? Do you have three-dimensional compasses? So we had to develop some methodology that allows us to record all 3 Euler angles, okay, yaw or azimuth, pitch and roll in a behaving animal. So we developed this non-coplanar array of LEDs that with 1 camera and 5 pages of three-dimensional trigonometry allows you to reconstruct the 3D Euler angles unambiguously while the animal is behaving. And we recorded from the presclaviculum, which is this region, sits in between the hippocampus and the frontal cortex at the back. This is, presclaviculum is the first area where head direction cells were discovered in rats 25 years ago or more. And so this is what Arsenian found. So first of all, this was published just a few months ago. So first of all, you see here examples of 3 neurons. And each row here is 1 session. So this is a 1 session. This is again a crawling session. This is in crawling. I'll show you a slide soon. The animal's crawling again 1 hour later. And so what you see here is pitch versus azimuth, and the color is the firing rate. Okay? So this neuron, for example, you see it doesn't care so much about pitch, but it's tuned to azimuth. So this is your classical azimuthal head direction cell, like in rats, if you will. But Arsenian also found neurons look like that, neurons that don't care so much about pitch, but about azimuth, but are tuned to pitch. This neuron, for example, likes negative pitch. You'll note, by the way, that this is limited to plus minus 45 degrees. That's because when animals are crawling, that's the behavioral range, right? It's crawling, so looking 45 up, 45 down, it doesn't breaking its neck like that. So I'll show you manipulations of 360 degree pitch soon, but this is what our behavioral range. And we also find neurons that are like this, that are tuned to a certain combination of azimuth and pitch. And in some rare cases, there are only a handful of these neurons, we have triply conjunctive neurons, neurons that are tuned to a combination of pitch and azimuth, roll and azimuth, and roll and pitch. But in general, tuning to roll was much, much weaker than to pitch. And I can tell you that out of the azimuth-tuned cells, the classic rat-like cells, about 35% were tuned to primarily to pitch, so we're conjunctive. That means that these kind of cells that encode three-dimensional direction in space are not that rare. It's more than 1/3. And instead, the tuning to roll was much weaker. Okay, then came the surprise. We did actually another session in the middle, which is having the animal inverted. Okay, so the animal is actually crawling upright, inverted, and then again upright. And these are examples of 3 cells, in which here is the azimuth tuning. So this neuron, you see, it's nicely tuned to azimuth, and an hour later it's tuned to the same azimuth. Okay, so this is stability. In the middle, it's still tuned, but to some other angle. Okay, this cell is the same thing, tuned to the same angle in 2 upright conditions, and to another angle in the inverted condition, and third cell, the same thing. And it took us 3 months to figure out what's going on here. I think some of you already know the answer in about 7 seconds. 7 seconds was the record. Somebody shouted, what's going on here? Anybody have a guess? Give you 7 more seconds. Okay, but the competition is tough. It will start off, and so the answer is that these 2 examples, 180 degrees. They shift up by 180 degrees, and they align. And in fact, it's not just these neurons, it's a lot of the neurons. So if you now look at the frequency in the preferred head direction for the upright versus the second upright case, the difference is around 0, which means the stability. But if you compare the upright to the inverted condition, the frequency, the difference in angle is around plus minus 180 degrees, so that's the inversion of the angle. And we interpret it as that the head direction is represented not in spherical coordinates, but in toroidal coordinates. So in toroidal coordinates, you have 360 degrees of azimuth and 360 degrees of pitch. Okay, so for a certain direction, all of this is the same azimuth. And this means that in toroidal coordinates, there's actually no 180 degree flip. It's all the same. Azimuth. And why would something like that be useful? Because if you think of star and spherical coordinates, the problem is that they have a singularity point in the zenith. If I'm a head direction cell like this direction, so I'm firing now, and you start to pitch, you still fire. You get to 89.9 degrees, you still fire. And then you do another epsilon, 90.1 degrees, you're supposed to stop firing abruptly. There is this discontinuity or a singularity point at the zenith, and that's non-biological, and certainly not useful for an animal that's flying and doing these maneuvers all the time, as I'll show you in a second. Whereas in a torus, you have continuous 360 degree of pitch and 360 degree of azimuth, so this problem is avoided. And indeed, if we compare the firing in the upright and the inverted conditions, they are more consistent under the 2 coordinate systems, they are more consistent with toroidal than with spherical coordinates. Then there were some predictions that I won't go into all the details because we're going to run out of time, but I'll say did some experiments in bats crawling on a ring. So now they in this kind of box where there was food here and also food there. So the bat basically volitionally chose to either go down here or down here. Basically crawl, then do a loop, crawl, do a loop, kind of on its own. So now we can compare with the same neuron firing down here and on the 360 degree pitch ring. And what he found is 2 things. First of all, that there's tuning. These are just 3 examples, but we have many more. That there's tuning to the entire unimodal tuning to the 360 degree pitch range. And the second thing that Dr. found is that if we now take a certain neuron, this is all one neuron, and we compare its azimuth tuning on the floor to the pitch tuning on the ring, we can kind of assume independence, multiply them, and get a model that looks like that. Okay? We didn't really cover the entire, experimentally, the entire 360 degree pitch range with 360 degree azimuth, right? We measured this and that, we multiply them, we get this. So now if you look at the experimental data, on the floor, which is limited to plus minus 45 degrees, as I just showed you, you see this is actually for that neuron very similar to a cut through this model. Okay, so this is actually supporting this toroidal idea. You can of course now close this line with that line and this line with this line, and then you'll get this toroidal representation. And this similarity between the actual data and this model is actually not just for this neuron, but for other neurons as well. This is the distribution of correlation coefficient across the neuron. Okay, so also I'll send you this flight experiment. I'm just showing you one slide and some examples that in flight it recorded, again, some cells were pure azimuth, some were pure pitch, and some were conjunctive. Like these examples, and the main difference between flight and crawling was the firing rate, as I just told you, was much higher in flight than in crawling.
That's right, in the lab.
It's in the lab, yes. In the same 6 by 5 meter room that I showed you before. So just to show you that this entire toroidal singularity argument is, or particle singularity, toroidal non-singularity argument is relevant, I'm going to show you some landings of bats on the ceiling. This slowed down 10 times. In reality, it happens 10 times faster, but you see that as the animal is landing right here, it went through the 90-degree pitch, through the zenith. Okay, so it's not just kind of a mathematical abstraction. It's a very relevant thing for an animal that is maneuvering a lot in 3D, and also when it's I can tell you when they're taking off, they're going through the minus 90 degrees. You'll see that in a second. So for the point is that an animal like this needs a continuous representation. Here the takeoff going to the minus 90 degrees. Okay, you got the point. So what are the features of the toroidal representations? First of all, as I said, continuity. Another interesting property is commutativity. And that relies on a very important point, that the animal should do little role. If commutativity is a difficult problem, as every theoretician, including Chaim, when 5 years ago I told Chaim we were starting a project on 3D direction scale, we had zero data. The first thing he asked, is it commutative, right? Because angles in 3D is generally non-commutative. So the answer is that the animals really, I'm not showing you that, but bats and a lot of animals are avoiding roll as much as they can. So here are examples of humans avoiding roll, all sorts of animals. If you look across literature from honeybees will never land, like, or Drosophila. We talked to all the experts in insect flight. They'll never do a coarse roll like this. They'll always land by pitch maneuvers. Animals try to avoid roll as much as possible. For pilots, roll is the worst source of vertigo, etc. So roll is really bad for you. And usually it's interpreted as you avoid roll to stabilize visual input, which might be true. We have another angle at this, literally, and that is that if you avoid roll and you have a toroidal representation in the brain, then the direction in the 3D compass is commutative, which is good. It makes computations much simpler. What happens in other animals, it's unknown, but even I, when I stand on the roof of a building and look down as a human, I know I'm on the roof of a building, not on the ground floor, so I have some sense of three-dimensionality. Show me monkeys and maybe other mammals. It's an open question what generalized for other animals. Mice, for example, as anybody who works with mice know, you go into a rack of mice cages, a lot of them will be hanging upside down. They're just fine hanging upside down, so maybe they have sources too in the brain. We don't know. Okay, in the last 3 or 4 minutes, I'm going to show you just a little glimpse of a very new and very exciting story, at least exciting to me. Very new data. That's the project by ELSC and Arseny Finkelstein that has to do with goal representation. So now with this new GPS that we have, we can record for, and this is the GPS recordings are done in collaboration with Yossi Ovet from Tel Aviv University, my former postdoc. So you can now record as an animal goes from the cave to a certain tree, and I'm just showing you some of the data. It's all data for one animal. It goes to this tree for many nights, about 3 weeks, I think. I'm just showing you some of the nights, and then it switches to another tree. Sometimes it's doing kind of choosing between them and then fixates on one. But some of the nights it's doing something like that. It's starting in one direction, then changing its mind and doing a shortcut to this other tree. Sometimes they'll do this. So we have, just in this animal, which is 75 nights, we have 5 or 6 examples of all sorts of shortcuts. So to me, this means they must have some sort of a representation of the goals. They know where the goals are. Now, how is that implemented in the brain? And there have been several suggestions. One is that you can do what's called pre-play of sequences of place cell activity in order to kind of plan your route of these distances. The problem is when you go with this idea, and there's evidence for that in kind of 2 meter or 1 meter environment. The problem with these scales is that we don't know how place cells look on these scales, but presumably you will involve many more neurons. It will be more complicated, and more importantly, we know that the rate of preplay is fixed, or kind of the meters per second the preplay phenomena unfolds is fixed. So if you want to preplay, you know, a route like this, you'll have to sit there for a minute or two. So there's certainly some possibility. So an alternative way, which I think is simpler, so we're going to test it, is to look for just a representation of goal direction. Just all I need to know, certainly if I'm a flying animal, if I want to fly from here to the Weizmann Institute at the end of this talk, sorry, no offense, but all I need to know is the direction of the Weizmann Institute, right? Then no matter what happens, I'll get there, right? So I use the representation of the goal direction, and then I think about a directional representation that it's angular. It means it's invariant to scale. So conceptually, it could be a nice way to do this. Nobody's found or looked for something like that in the brain. So to answer this question, we are now running experiments. Ed and Arsenia are running experiments where the bat is flying in our flight room. There's a goal here, a platform on which it lands. And then we're asking ourselves, okay, if this is now the heading direction of the animal, and this is the direction to the goal, the difference between them is what we call the goal direction. Okay, now you can ask, when the goal direction is 0, so the animal is going towards the goal, does the neuron fire at a higher firing rate? And for these 3 neurons, the answer is yes. What you see here is the representation, it is the goal direction angle, and this is the firing rate. You see that they're tuned to 0. Okay, it's important to understand this is not head direction. It's not a compass. It's not north. Okay, the goal direction is fine with my goal. Goal direction will be 0 like this. Okay, but it will also be 0 like this. Okay, so it's not absolute direction in space. It's really direction relative to the goal. And so these 3 neurons are tuned to 0 goal direction. So they encode the direction to the goal. There are also some neurons that are tuned to other angles, not to 0. And if you look at the distribution, these are preliminary data, so certainly we shouldn't take the distribution too seriously. There seems to be a little overrepresentation of 0 angle, 0 direction, and 180 degrees, so like where you're heading and where you came from. So, and this is not due to, this cannot be explained by place tuning, because if we now look at the place tuning of for example, this neuron, we can ask, how is the place tuning inside the place field and outside? And you see it's reasonably similar. If you compare now for that neuron, you look, take the spike inside the place field, inside the gray area, and outside, the place tuning is the same. The goal direction tuning is the same for this neuron as well, for that neuron as well. We can now ask, how is the goal direction tuning inside and outside the place field compared across all the neurons that we have so far? And it's around 0. So it's not really stemming from the place tuning of the cell. Some of them have. And finally, the goal direction tuning is stable across time. This is, surprisingly enough, hippocampal area CU1, which is the second most studied area of the brain. So last week, I think we now have serious competition, I must say, because last week Arsenia and ELSC presented this in the hippocampal conference in Italy, and everywhere, this was the big buzz in the conference. Everybody did not believe this happens, and they're not all going to now look at this in their data, because a lot of people have CU1 data in rats, so we should hurry. Yes?
If I understood correctly, if you want to update your angle towards the goal where you're moving around, you do need to represent also how far the distance.
You're right. So in order to create this representation, you need to know something about where you are. And I don't have a good answer about how it is created, but
you should expect to have some representation of that form somewhere.
Yeah, right. No. And I can already tell you that there are more place cells in C1 than these goal direction cells. It's not like place cells are wrong, it's just that there is this extra signal. If you think about this, just another kind of signal in the hippocampus that tells me something about where the goal is. Let me just see we're running late. I'll just take 3 minutes to finish and then open for questions. So this just shows that this representation is stable over many meters. It's not just when you approach a target, but many meters before that along the path. These 3 example neurons are tuned to 0. So really, it's a stable representation. So we found here in preliminary data a subpopulation of hippocampal neurons that exhibited rather narrow angular tuning to goal direction, and this could support goal direction navigation because it's really straightforward to decode this in order to get to a goal. So future plans, we're working on genetics in bats. Viruses work, and I won't say much about this yet. And major other future plan, and it's more like present plan right now, nowadays, is asking this question. Is everything I told you about 1 meter boxes relevant for the real world? So to answer this kind of question, you need to develop technology. So this, maybe first I'll for the technology about the science. So why is this a non-trivial question? And the reason is that if you just think about what I told you about place field, if you want to take this 1 meter place field and you want to tile a 20-kilometer flyway, which is 2 kilometers wide and half a kilometer high, you'll end up with 10 to the 12 neurons. If it's smaller place field, maybe 10 to the 15 neurons. That's 1 billion times more neurons in the hippocampus. That cannot be the case. This cannot be the case in the same way that it works in 1 meter boxes will work in the outer world. So what are some options? One option is that you'll have a hierarchy, meaning hierarchy of resolution, meaning let's say inside the cave and around the trees you'll have small place fields, high resolution, and up in the air where you really don't need a 10 centimeter resolution, you have, let's say, a 300 meter large place field, right? It makes sense, but nobody looked for that. Another possibility that was predicted by Ketch and Zhang is that you have hundreds of place fields, small ones, but hundreds of them. It's like hundreds of receptive fields for a V1 neuron, and he argues this based on all sorts of optimal decoding ideas, unpublished work by Ketch and Zhang. So there are very different contrasting ideas out there for what will happen, and really need to go and look for that. So to answer this kind of question, we're developing now, or developed already, this neural logger. So this is a device that stores the data on board the animal. Now it's not transmitting it by RF as before, because that's limited in range. This thing is storing the data on board the animal. It weighs 4 grams. So think of your rack of 20 kilos of electrophysiology rig. That's 4 grams doing the same thing. Really, it's the analog front end, digital, the computer, the memory, everything's right there. 16 channels, 4 tetrodes, and stored on board the animal. And we get nice clusters, and we're really now starting to run experiments on large scale. Hopefully in the next year we'll have some answers to this question. So this is my summary slide. As I promised you, why do we study bats? So first of all, they have temporally discrete sensory input, this sonar, which are interesting if you care about let's say active sensing. But for me, the important thing, I can measure the timing of sensory input with a 1 millisecond precision, which turns out to be very important to affect these neurons. I didn't show you that. I published a paper showing that there's an interesting effect of the precise timing of the sensory input on activity of place cells in a rat that runs in an arena in the environment. When exactly does it touch something, see something, smell something? It's just a measurement problem. So there's an advantage for bats there. 3D, of course, theta oscillations happen to be different in bats, so you can constrain the theory, space of possible theories. Excellent spatial memory, want to bridge the gap between behavior in the lab and outdoors. And the two most important points for me are this comparative systems neuroscience point. I really think that for sensory systems, people have looked across many species. What's comparable and not comparable for vision, audition primarily. But in the hippocampus, for historical reasons that I understand, but I don't agree with, 99.9% of what we know comes from just one species, the rat, and a little bit from the mice. And I think at least as a sanity check, we need to look at other species, what is invariant, what isn't. And I think we should focus on the invariant properties to understand the system. And secondly, this idea of natural behavior. I'll be happy to argue why, if you want to understand vision in mouse, you need to understand the natural context in which it uses vision. There are examples of why this is important. And in any system, this will be important. In our system, the navigation system, you want to look at natural behaviors, goals, large scale, complexity, etc., in order to understand the system. Okay, so I'd like to thank the lab members who bravely explore 3D. This is from a lab trip that we did in September in the Black Canyon of the Zavitan in the Golan Heights. And here, Seni and Alon are checking what happens when you go beyond the zenith. And Tamir, I'm not sure exactly what they did here, but they're kind of pushing the limits of 3D, that's for sure. And I'd like to thank all lab members, in particular the overlap, who was my right hand in the lab, and many of you know her, she's really fantastic. And the students, I think I mentioned in just pictures of most of them, or all of them actually, and collaborators, alumni, funding sources, and thank you for listening.
A few questions.
And what about theta sequences if you look for something like that?
Theta sequences. So we don't have typically enough neurons per recording because we opt for very lightweight microdrives. They can carry a 12 tetrode microdrive, but then they don't fly as well. I prefer to have 4 tetrodes and they have them fly really well, but this means we have maybe up to 10 neurons simultaneously. 5 to 10 would be a good day. Simultaneously, so it makes it difficult to do the data sequences. What we can do, and we're looking at that, are pairwise correlations. Because we have a lot of pairs of simultaneously recorded neurons, and there are some interesting phenomena there. Correlations getting stronger after behavior as opposed to before behavior. So things like Wilson, McNaughton saw at the time. So something we're looking at now.
Regarding the goal direction, did you try to have the goal be hidden so that you will not see this and use memory?
Yes. So the surgery we're doing on Monday is for a bat that is going to fly and have a task where there is a hidden goal. So what Udi is asking about is about a hidden goal, and it's important because I think it's a very basic question. Is it a visual representation or a mental representation, right? So we have now a goal that's in the open environment, but we have a curtain, and there's a goal behind it that's in the various positions, and now so the animal flies behind the curtain, it knows, and when it's here, it knows the goal is there, but doesn't see it, right? So is it a mental memory-based representation or sensory representation?
What a maze for the rest.
Yeah, so we've done a little bit preliminary experiment like that already in one animal, and there seem to be cells that are doing that, but now we're going to do this next week much more systematically. But I think it's very good and very fundamental question. Yeah.
Yeah, I was actually a related question about obstacles.
Yeah.
You have a straight line, but sometimes you have to maneuver around obstacles.
Yeah.
And do they come back to the straight line?
Yes, and certainly if you put obstacles, they will go around them. People have done behavioral experiments in that years ago. You put an obstacle, they'll develop a detour, then you remove the obstacle, there's still be this detour for a little while until they figure out it's not there. So, animals, humans do the same actually, think about it. It's habits, it's not really hippocampal dependent. But we haven't looked at the brain with this kind of manipulation, but we're planning to do this in the next year. We need to build something bigger, which we're working on now, and then we'll be able to do
Because you talk about natural movement, that's very natural.
Okay. So you need big spaces to do that, and hopefully it's already in construction, so then we'll have an answer. Yes.